Welcome to solverfy.
This question asks, what technique is used to increase the number of DNA or RNA fragments? And the answer is PCR, or polymerase chain reaction. And the way PCR works is that you have two different, let me do that a little bit lower, two different DNA fragments. This is fragment 1, fragment 2. And this is the 5' end and 3' end. So, of course, on the other strand of DNA, this is the 5' end, and this is the 3' end. This DNA is then heated up so that it denatures or not denatures, but so that it separates. So you have the strand here, and then, or why don't I do this on a new page? It'll make it a little easier. So after the DNA is heated up, you have this strand, and then you see it's separated. And here's the other strand. And again, here's the 5' end, 3' end, 3' end, and 5' end. And after the DNA is heated, it is then cooled down so that the primer can then stick onto the DNA. So let's get the primer. So let's say that this is the primer. And 6 DNA. And what the primer does is it then synthesizes a new strand of DNA. And we know that DNA is synthesized from 3' to 5'. So this primer will go this way, and this primer will go this way, 3' to 5'. And then what you end up with is two completely different DNA strands. So you have this DNA strand, and then you have this DNA strand. And then this is 5', 3', 3', 5', and then the exact same thing here. So you can see using polymerase chain reaction how DNA and RNA can be amplified. 3' and 5'. This is a 3. All right, so the answer is D.